Good evening, everyone. So in the next few minutes, we are going to talk about some of the scientific advancements and the emerging technologies that are being developed at AgResearch. For example, cold plasma, dual light treatment, and hyperspectral imaging and deep learning, which is more of a detection technology. Now, in general, there could be several ways to define what an emerging technology is, but they all are potential advancements which are pre-commercialization stage, and they promise to deliver beyond what conventional technologies do, and they have sustainability goals and ensure food safety and shelf life. Let's start with cold plasma. So cold plasma is also known as the fourth state of matter, and this is generated by ionizing gas. So the gas composition should be known in most of the cases, and depending upon various parameters that could be controlled, we can generate reactive oxygen and reactive nitrogen species in this gas. And that gas, when used to treat water, generates plasma-activated water, which again has these reactive species in there. Now, these species are active against bacterial cell. When saying that, there would be a species-dependent effect, for example, gram-positive, gram-negative, spore formers, et cetera, et cetera. And it's at very early stage of research, I must say. So I'm trying to present case studies for all these three technologies today. We'll start with this one, where we tried using plasma-activated water on meat coupons, just to see what happens to the natural microflora out there. So we used meat coupons and washed them with plasma-activated water, and control was our deionized water. After washing it for about 5 minutes, we left it in the refrigerator for 12 days and monitored aerobic and anaerobic counts. And as you can see, plasma-activated water kept the counts low by 1 to 2 logs, up to 12 days. What is the next step was challenge testing. So we tried using Salmonella and E. coli on meat, so inoculated up to 6 logs, and the plasma-activated water generated was by activation of 8 minutes, but we incubated these meat coupons, inoculated with these bugs, in water for about 30 minutes, 1 hour, and 2 hours, just to see what happens. And you could see that it did work. It brought the numbers down by 1 log colony forming unit, but that's it. What was more interesting is these changes on the bacterial cells. So these are the scanning electron micrographs on Salmonella typhimurium cells, and you could clearly see there is damage on the surface of the cells there, which is promising. Similar sort of structural damage could be seen on E. coli as well. So the work is in progress, and we'll know more in the coming few months or years. The next technology we are investigating is the dual light treatment, which involves using blue LED and far UVC together. Now, the initial proof of concept was led by Dr. Gail Brightwell, and that paper was published, in which the dual light setup proved to be very effective against SARS-CoV-2. The similar setup, as you see in the photograph here, is being used against bacteria now, and we tried ESBL producing E. coli sensitive, as well as resistant strains were used for this trial. Now, there were 2 research questions here. One is, would the dual light improve in activation of E. coli? And the second is, since there's UV involved, we wanted to make sure it doesn't alter the antibiotic sensitivity in any way. So this paper has already been published, and a lot more information would be available in there. Now, just in a summary, we found that dual light treatment gave better reduction by equivalent of 1 log colony forming unit per ml. And here, dual means when both the treatment was together, whereas combined means adding the log reduction obtained by UVC and plus the blue LED. So using dual sort of gave a better inactivation effect, and the mechanism is under investigation. We saw that the antibiotic sensitivity profiles do not change even after many passages and many subcultures of the treated cells. Now, the third technology is hyperspectral imaging for detection and quantification of bacterial spores in food. So we have multiple projects on this, but I'm going to talk about my proof of concept project, which was on Clostridium sporogeny spores in food. So the research question here was, would hyperspec imaging be able to detect between live and inactivated or non-viable spores? And second question, if it does, can it quantify the spores? So we used two types of matrices. One was media plates, and the other was food model, which is mashed potato in this case. Now, that paper has also been published, so there's more information available. Now, in summary, the hyperspec imaging just through PCA plots indicated clear clustering difference in live and dead cells, or a combo of both, when plates were used. But the same thing when applied on food, that is mashed potato here, it was a mess. So there comes the next step, which is the data processing, which was using deep learning approach. Now, I'm not an expert of this, but I have collaborated with the artificial intelligence experts for this purpose, who could be contacted if you need to. So two types of models were tested, random forest model and 1D convolutional neural networks. And we found that both these models were very good in detecting dead and live spores, spores in the presence or absence of non-viable or spores could be quantified very well. In general, the technique is to detect all the biochemical peaks generated by the mashed potato and then masking that in the actual samples. In other words, training the system to be able to pick up what is needed and not detect what is not. This is how it looks in general. So the system was able to tell us red and blue, dead and live cells very easily. However, as usual when it comes to quantification and as the numbers go down, it became more and more difficult. So the accuracy was 98% if you're just asking it to tell you dead or live, but it reduced as we asked it to quantify the number of spores, and it further reduced when there were dead cells present. So our current projects are looking at strategies to mitigate these sort of confounding effects of the background. These are the projects underway at the moment. So we are looking at optimization of irradiation, cold plasma, etc., on pathogens of interest. And we'll soon have a PhD project on plasma and disinfection of offal meat with University of Otago. And there is another project on the effect of dual light treatment on bacterial spores using the omics approach. And I'm also leading the New Zealand-China Food Protection Network project, which has a postdoc looking at metabolomics to help to mitigate the confounding effects of hyperspec imaging. Last but not the least, I'm trying to use hyperspec imaging to monitor Bacillus cereus spore germination in food. That's our whole team, and I thank everyone, and thank you for the opportunity to talk today.
Thank you, Aswathi. It amazes me that the range of expertise microbiologists is expected to master these days. I'm fortunate I grew up when all we had to do was just, count the numbers on an agar plate. Anyway, do we have some questions for Aswathi? Pierre.
Thank you very much. It was a really nice presentation. Just on your plasma-activated water, you said as a control you used, was it distilled water or deionized water?
Deionized water, yeah.
What effect do you get if you just use standard chlorine-based, chlorinated potable water? What's your log reduction then?
I haven't tried that yet because the whole idea is to avoid chemical disinfectants and try to go green. But another research questions that we currently have is to look at the food-grade chemicals and plasma activate that to see if it improves the efficacy. So probably I'll have an answer soon.
I guess the follow-up then would be, are you also working with a regulator to change the regulation? Because if it's food contact, it needs to be potable water, which means it needs to have certain levels of chlorine, etc. in it.
Yes, the research is at very early stage at the moment. So if it's promising, definitely regulators would get involved.
Thank you.
Thank you.